Nowadays, environmental problems have become one of the main global issues. This kind of problem may come from various sectors. But did you know that there is one precise sector that may create an unexpected impact on our environment? It's gold mining.
In many countries, artisanal and small-scale gold mining, or ASGM, has become a working field among communities. However, there are many challenges to create sustainable non-mercury gold processing. One of the main materials widely used in gold mining is cyanide, which separates the gold elements from the traces. However, its use may leave harmful waste that may cause massive kills of various organisms and even threaten our health. With its limited performance of resources, ASGM also widely utilizes cyanide as a leaching agent in the hydrometallurgical process. However, there are many occurrences of cyanide leakage and improper waste treatment within its practices. As a result, this treatment is unsuitable for ASGM. Starting from those problems, we aim to discover a better way of cyanide waste treatment using biohydrometallurgy or bioleaching.
Coming with the concept of synthetic biology, we are trying to develop an engineered Chromobacterium violaceum to have a proper cyanide regulating system, resulting in a better gold bioleaching and cyanide waste treatment. So, this is our project, AuViola.
The AuViola project uses bacteria called Chromobacterium violaceum, a non-sporing gram-negative bacteria, to take over the gold bioleaching process. This bacteria has the ability to produce cyanide by expressing the HCN-ABC operon. This bacteria is engineered to optimize the cyanide production and regulation to both improve the yield of gold bioleaching and to prevent and minimize the negative impact of hydrogen cyanide leak in the environment. We are trying to design an inducible pathway which can degrade the hydrogen cyanide after it is used. Through AuViola, we are constructing an on and off system between HCN production and HCN degradation.
We designed a cyanide production circuit which consists of the ROC-PBAD promoter, strong RBase, HCN-ABC operon, which we synthesized from Swiss Bioscience. However, for the HCNC, we directly isolated from the Chromobacterium violaceum due to the high GC content. And lastly, we also need the double terminator to complete our parts. Both of our parts were assembled using gypsum assembly and transformed it into Escherichia coli DH5-alpha and also BL21DE3. In order to confirm our gene construct, we do electrophoresis and send the isolated plasmid for sequencing. Aside from that, we also characterize this part into BL21DE3 and also DH5-alpha to know the level of expression of the GFP protein and also the growth curve.
For the dry lab, we designed various models such as kinetic modeling, genome scale modeling, and bioreactor system. The first model, kinetic modeling, consists of cellular scale, HCN or hydrogen cyanide regulation, and gold cyanidation. The HCN regulation comprises of HCN production, HCN degradation by rhodonase, and arabinose conversion by L-arabinose isomerase. These three frameworks combine to create a comprehensive scenario of every molecule concentration per time. The second model, genome scale metabolic modeling, predicts the gene knockout to enhance the flux in cyanide production metabolic pathway. Then, we compared it with the wild-type metabolic map. The bioreactor system is the last model that answers the possible real-world application of our project. The system must be suitable to extract refractory gold, where the gold atoms are trapped between other minerals.
For the results, we successfully isolated all of the desirable construct either from BioBreaks or the Chromobacterium violaceum genome. After the transformation and random picking colony PCR, we obtained the unexpected result where all of the colonies merely have around 1500 base pair size of amplified gene. However, for this sample, the Col-A sample showed thin bands with the size of around 4000 base pair. After we subcultured this colony, we obtained similar bands on the gel electrophoresis with the size of around 1500 base pair and 5000 base pairs. Thus, we decided to just send it for sequencing for further confirmation. However, we are still waiting for the sequencing result up to this day. We incubate the BL21, DE3, and DH5 alpha clones in the LB medium, which supplemented with chloramphenicol. And then we measure the growth curve and the GFP expression with the addition of L-arabinose as the inductor. And here is the result of the growth curve. It is apparent that both clones showed lower growth rather than the control. This result suggested a higher metabolic version of the clones. The foreign genetic parts expressions demands more cellular resources and energies which compete with the native host metabolisms. Therefore, the host cells may suffer from delayed growth at the beginning. On the other hand, the measurement of GFP expression depicts that the BL21-DE3 experienced significantly higher expressions of this protein rather than its counterpart, especially after the induction of L-arabinose. However, the GFP expression was not detected in both of the control strains. It revealed that the DH5 alpha strain did not specialize for the heterologous protein expression. And here are the documentation which shows the visualization of both cell pellets after 8 hours of incubation after the UV trans-illumination. By the time you receive the sequencing results, we will directly assess the construct and measure the accumulation of cyanide production in Escherichia coli BL21-DE3 expression system. By contrast, another assembly and reconstruction will be conducted when the result were not as we expected. Besides, we also plan to change the origin of replication from PBBR1 MCS2 plasmid because this origin of replication is compatible for both Escherichia coli and Chromobacterium violaceum. After that, we also plan to measure the cyanide production in Chromobacterium violaceum after we transform our engineered plasmid into it. For further improvement, the pH optimization is needed because cold cyanidation is a pH-dependent process. In addition, for the safety consideration, we try as best as we could to prevent any leakage of every engineered bacteria to the environment. We performed detoxification of all tools and equipment that we used. Besides, we also protect ourselves during the wet lab work with protective wear, such as the lab coats and single-use gloves.
To model the system, we need to divide HCN or hydrogen cyanide degradation into two parts. The first one is rhodonase production, and the HCN degradation by rhodonase. We also divide L-arabinose conversion by L-arabinose isomerase enzyme into two parts, the enzyme expression with weak Anderson promoter and arabinose conversion by the enzyme itself. Some of the reactions are really fast or almost spontaneous, such as binding of arabinose to RC complex to operator, HCN synthase transport to membrane, TET-R dimerization, HCN dissociation, etc. Those reaction effects were assumed to be negligible. In addition, some molecules are assumed to be constant all of the time. For instance, arabinose concentration inside a cell, RC complex, etc. Therefore, with all of that assumption, we could shorten the overall reaction and make it easier to model our frameworks. HCN or hydrogen cyanide spikes to their maximum concentration in the first 60 seconds, then decrease slowly because it starts to react with gold. L-arabinose is consumed by cells, and when it reaches 50% of its initial concentration, rhodonase will be produced. The steeper HCN graph proves that rhodonase contributes to the reducing of the HCN concentration. We separate the model for this reaction to see the effect of HCN versus gold without rhodonase interception. We assume that the gold particles are totally liberated, sphere, and in abundance. For reactions, we assume the reaction happens on the surface and follows a shrinking core model. Turns out, the potential gold recovery keeps rising before the cyanide runs out.
To enhance the yield of gold cyanidation, the cyanide concentration produced by Chromobacterium violaceum should be optimized. One of the solutions is by performing the knockout gene to sort up the flux of cyanogenesis pathway. Therefore, we decided to do genome scale modeling to level up the cyanide production in C. violaceum. After doing some modifications and adding some reactions to the map, then we tried to optimize the cyanide production. First, we perform a constraint-based method which provides a global view of how optimal growth rates are affected by the production of cyanide.
We also predicted the knockout strategies using the opt-in and opt-knock, which is provided in Cameo to find out solution area through the production envelope. In addition, we also compared the reaction flux between the wild-type and mutant C. violaceum metabolic maps, especially in cyanide production pathways. The phenotypic phase plane provides us a global view of how biomass is affected by the production of cyanide. This area shows us the trade-off between our target, in this case cyanide production, and the biomass. We used Chromobacterium violaceum manually created biomass encoded by biomass 1.12 to define growth, and the reaction glycine acceptor oxyreductase encoded by RDB00166 to define the objective of cyanide production. Based on the prediction of knockout strategies using glycine acceptor oxidoreductase as the target reactions and the glucose exchange as the initial reactions, we found that there were 3 knockout SIMs from the results of opt-in, and 2 SIMs were obtained from knockout prediction using opt-knock. One of them is the knockout of ammonia transport reactions. Based on the simulation result, the production envelope from the knockout prediction using opt-in was the same as the result with opt-knock. This production envelope depicts the solution area from a constraint-based method. Based on this solution area, we could imply that rewiring the flux to produce cyanide will impact to the lower growth of C. violaceum.
Both the knockout prediction using opt-in and opt-knock provide the top solution in the form of knockout chains of the ammonia transport reactions. Based on the flux analysis, it was seen that there was an increase in flux towards cyanide production in the opt-in and opt-knock mutants compared to the wild type. The targeted reaction, glycine acceptor oxyreductase, experienced flux increment slightly over 20 times. This increment is followed by several flux changes in other reactions, as you can see here, which overall leads to the glycine acceptor oxyreductase present flux increment. This free phosphoglycerol shipment to the cyanide production pathway may also be related to the plummet of cell biomass on the production envelope graph that has been shown before. In addition, based on previous study by Tay et al. 2013 mentioned that the cyanide production pathway is a branch from the conversion of free phosphoglycerate, which is an intermediate in glycolysis.
In the knockout prediction, it is recommended to use an exchange reaction as the target of knockout strategies, because an exchange reaction can represent the accumulation of targeted metabolite in the outside of the cell. However, in our knockout model, we use glycine acceptor oxidoreductase, which is a cyanide production reaction, as the main target, because the cyanide exchange reaction is still incomplete in our model. Therefore, we suggested it is necessary to improve and add the cyanide exchange reaction to the model and transform the target in the knockout predictions into the cyanide exchange reaction.
We designed 2 types of bioreactor systems, one that extracts gold from refractory gold ore and one from non-refractory gold. Refractory gold needs one more reactor for breaking the surrounding minerals before gold cyanidation occurs. Gold atom in refractory gold is surrounded by other minerals, and it is almost unbreakable by cyanide alone. Based on clone refractory gold ore, we designed the first bioreactor with Thiobacillus ferrooxidans to dissolve the pyrite minerals around the gold atom. Thus, with bioreaction kinetics parameters gained from various research, we modeled the reaction over time. In this graph, we can see the cell, product, substrate concentration, and volume addition over time. Also from this graph, we decide the batch residence times is 4 hours, and the total volume is 30.24 liter, with 20% over design considerations to prevent reactor flooding. We utilize a heater after the reactors for the bioleaching process to kill the bacteria before the stream entered the new system.
Our mission as human practices is to connect and engage with small-scale gold miners and listen to their experiences and concerns. In addition to that, we have conversations with the stakeholders and experts to understand the bigger picture of artisanal small-scale gold mining waste problems.
Finding a solution to the cyanide waste problem could be one of the keys to mitigate cyanide pollution and poisoning, especially around artisanal small-scale gold mining or ASGM area. To achieve it, we have to make sure that our solution is right on target, cost-effective, and also problem-driven. We have formulated the human practices design to be able to make an organized way of thinking to close the loop of ASGM waste problems using synthetic biology. Our integrated human practices framework reflects on our activities such as literature review, interviews, and the sharing sessions to understand current problems in gold mining activities. From those problems, we created some reflections as our guide to formulate and build the solutions. Furthermore, we aim to propose implementation and future improvement that can be initiated from our project. The first step is understanding the problem. To find out the gold miner's point of view, we visited one of the ASGM that is still operating in the Kulon Progo district, special region of Yogyakarta, Indonesia. We know that the current challenge is the difficulty in the obtaining cyanide raw materials and its complicated waste processing. On Monday, the first week of August, we held an online sharing sessions with the Gold Ismia project team. Coming with the session, we aim to deeply learn about the current application of mercury in Indonesia's ASGM, as well as explore the benefits of cyanidation in the gold mining process. We also interviewed Professor Suhadi, an expert in the environment, to find out that the levels of HCN content in the soil and water around the ASGM area at Sekotong district have exceeded the WHO threshold. The soil and plants are regarded as polluted and create a negative impact on organisms living nearby. This is due to the habit of throwing away the cyanide waste without further processing to the environment. He also said that a new practical alternative approach is needed in order to save the environment. From the literature review, community engagement, expert consultation, and brainstorming, we got the idea to make a tool using synthetic biology, which can produce measured amount of cyanide and eventually degrade it into less toxic materials. To also ensure that our method is profitable for artisanal small-scale gold miners, we created an economic measurement model. There are two variables that can be measured, CAPEX and OPEX. We compared our model to the existing conventional method and found that our proposed model will require less energy, less LPG, and less money. In addition to that, we had done several other human practices such as education and communication, collaborations, and also partnership. On August 27th and 28th, we held an online event entitled UGM Sinbio Online Festival 2021. Our main goals in organizing this event were to introduce the synthetic biology and iGEM competition as well as promote our AuViola project to Indonesian students. This event was also attended by other iGEM teams from Universitas Indonesia, Universitas Brawijaya, Moscow City, and Korea HS in order to provide various perspectives as well as create a comprehensive collaboration. Being held for 2 days, the UGM Sinbio Online Festival consisted of 3 sessions. First is webinar, second is discussion, brainstorming, and hands-on in silico plasmid design. Third one is mini talk show, Meet the iGEM. On September 11th, we were glad to participate in the Kodiathon, held by the Manchester 2021 iGEM team. At this event, we spent some of our quality time working on wiki pages for 24 hours. Although we had to deal with 7 hours time difference, we were happy to also meet other participating teams from ISER, Trinupantapuram, Warwick, Franzymes, and Inhoven in its every 3 hours check-ins. During partnership, we received primers and L. arabinos from the iGEM Brawijaya team in return for BL21DE3, as well as linearized plasmid backbones for the PSB1C3 and PSB1K3 plasmids that iGEM UGM Indonesia sent. Without the help of the iGEM Brawijaya team, we wouldn't be able to continue our experiments. iGEM Brawijaya also recommended us to use Gibson assembly instead of using restriction and ligase enzymes.
Implementing AuViola in the real world will take a long journey. We realized that we were unable to complete all sections of this research at this time. In the future, we are planning to divide this research into 4 phases. At this moment, we have taken the initial step in gene construction and dry lab modeling. We consider this as the first phase. In the second phase, we need to optimize HCN production of Chromobacterium violaceum. In the third phase, the focus will be on the HCN degradation system. The last phase is to build a real bioreactor and create the best adjustments that can produce the best yield of gold, yet minimum waste products. However, the solution that we offer needs to be integrated with various stakeholders, NGOs, industry, and open up opportunities for research collaboration in the future. According to the serious damage caused by cyanide, we hope that AuViola will provide a better environment for the living organisms, for making a world of difference to the gold bioleaching process, and also drop cyanide poisoning prevalence or incidence, resulting in a better human health.